Okay, um, I'm David Pellman, and it's my absolute pleasure to introduce Jan Lammerding for our Seminars in Oncology series this week. Um, Before I say a couple words about the lecture today and introduce Jan, just a couple of business items. After the lecture, I'm going to ask people to um, uh, post their questions in the chat. Hopefully we'll be able to get through many of those questions, but we'll forward those to Dr. Lammerding and he can hopefully answer some of them by email if things come up afterwards. So this is one of our special lectures today. This is the Charles I. Siegel Memorial Lecture. And this lectureship was established in 1989 by Charles and his wife, Edith Siegel. They're longtime supporters of the Jimmy Fund, Dana-Farber, and other charitable organizations in Massachusetts and Florida until their passing. Their only desire for this lecture is that it continue to educate future generations of medical professionals and those that attend the lectures. And they send the following encouragement to us, which is to take notes and study hard. So I myself know that I'll be doing that because we have a wonderful speaker today. Jan Lammerding is a biophysicist and a biomedical engineer who is tackling some of the real cutting edge problems in cancer biology today. Jan was an undergrad at Dartmouth and then did his PhD in biomedical engineering at Massachusetts Institute of Technology. And then he came for a postdoc with Richard Lee at the Brigham and Women's Hospital and stayed on at Brigham as an assistant professor until 2011. In 2011, he moved to the biomedical engineering department at Cornell and rose through the ranks, and he is currently now a full professor and the head of the graduate program at Cornell. Jan is known for both technical and conceptual innovations and on the mechanics of the nucleus and its role in cell migration, the control of gene expression, and genome stability. I could go through a lot of his fantastic discoveries, but I'm going to cut this off now and let him take the floor, give him enough time to speak and hear in his own words. So thanks everybody, and again, post your questions at the end in the chat.
Great. Thank you very much for the kind introduction, David. It's my pleasure, I guess, being back in Boston, even though it's just virtually. It's just unfortunately not physically here, but hopefully some other time. And can everybody see my screen and hear me well? Good. So as David mentioned, we study mechanobiology of tumor cells. Overall, our lab works on two large research areas. About half of our efforts is dedicated to understand how mutations in nuclear envelope proteins such as lamins cause heart disease, muscular dystrophy. But then the other half of our lab, and this is what I'll be talking about more today, really investigating how the structure composition of the nucleus can modulate and influence the behavior of tumor cells, and conversely, how mechanical stress can affect the function of these cells as well. To give just a brief background on the nucleus itself, of course, I think here everybody's familiar with what a cell nucleus looks like, but I think the impression that you often get is by looking at textbook or even your histology slides, you just see the nucleus as a big blue blob in the center of the cell, knowing that there's the DNA in it. But I think what's less apparent but important to point out is that the nucleus is really physically connected with the rest of the cell. And so as you can see, in this slide here, we have the nucleus. We have a structure that's called the nuclear lamina, which is underlying the inner nuclear membrane. But then we have proteins such as sun proteins and nesprins that connect the nucleus, the nuclear interior, to the cytoskeleton, and via the cytoskeleton to the extracellular matrix or to other cells, meaning that mechanical forces that are either generated inside the cells or on the outside of the cells are transmitted all the way to the cell nucleus. And if we look at some of the physical properties of the nucleus, it's important to recognize that the nucleus in most cells is the largest organelle. So it's typically on the order of about 3 to 20 micrometers in size, which means that in most cells, it really occupies a large fraction of the cell volume, as you can see in this electromicrograph of a human fibroblast. In tumor cells, we know that it's often a pathological feature that the nucleus is enlarged and has abnormal shapes. It occupies an even larger volume of the cell. What's important to recognize as well is the nucleus is not only the largest organelle, but it's also the stiffest organelle. So it's about 2 to 10 times stiffer than the surrounding cytoplasm. And so if you think of the combination that the nucleus occupies a large volume of the cell, but it's also much more rigid, you can imagine that the overall physical behavior of a cell, how a physical cell is deformed under force, will depend much on the structure and mechanical properties of the nucleus. And so what do we know about the mechanical properties of the nucleus? We know that they're mostly determined by the properties of the nuclear lamina, this dense protein network underlying the nuclear membrane that you can see here in this micrograph as well as the nuclear interior, so the chromatin, DNA, and DNA binding proteins. And just as a very simplistically speaking, you can think of the nuclear lamina as a mostly elastic network, almost as like a rubber shell around the nucleus. And then the interior is what's called a viscoelastic material, so it shows time-dependent behavior. So it's like a honey-like gooey substance in the nuclear interior. And so this is really just the very simplistic view of the cell nucleus. If we're trying to understand what role specific lamins have, so we know that mammalian cells express A-type lamins and B-type lamins, lamin A and C being the main A-type lamins, we know it's really these lamins A and C that determine the deformability of the cell nucleus. These were some experiments that I did just down the road, I guess, from where you are now as a graduate student, where we were stretching cells that are deficient for specific lamin proteins and measuring the nuclear deformation. And what we could clearly see is that in a normal cells that has high levels of lamin A and C, the nucleus is very rigid and it does not deform a lot, as you can see here in the top right image. So even though we stretch the entire cell by about 20%, the nucleus deforms very little. In contrast, if you look at lamin AC deficient cells, we see that the nucleus stretches more, it's much, much more deformable. While if we delete B-type lamins, lamin B1 or B2, the stiffness, the deformability of the nucleus does not deform much. So you might wonder, well, what does that have to do now with cancer cell biology and metastasis? I think I don't have to tell the audience here that we know that metastasis is, of course, very important. It's responsible for the large majority of of cancer death. We study primarily breast cancer, and so we know it really is just the main driver of death in these patients. That part has been known for a long time and motivating cell migration research. However, what's only become more recently into the forefront is really the physical challenge that cells have to undergo as they metastasize, as they spread from the primary tumor through the interstitial tissues to reach blood vessels, enter blood vessels during the intravasation, exit blood vessels during the extravasation. And importantly, during these processes, the spaces that the cells have to crawl through are typically on the order of about 2 to 20 micrometers in size. So on the same size or substantially smaller than the size of the nucleus. And given that the nucleus is the largest and stiffest organelle, we've been wondering what role does now do the physical properties of the nucleus have on the ability of cells to navigate such spaces. And just to really show you how physically challenging is the cell migration through such three-dimensional environments is, these are some latest light sheet images that we acquired at the HHMI Advanced Imaging Center at the Janelia Research Campus, where we have breast cancer cells, MDA, MB231 cells, where the nucleus is labeled and the actin cytoskeleton migrating through a collagen matrix. And the collagen matrix, you cannot see it, it's not fluorescently labeled, but it has pore sizes on the order of about 2 to 5 microns, so smaller than the size of the nucleus. And you can clearly see on the left-hand side, where you have a cell that is not migrating much, you have the typically expected oval shape of the nucleus. But on the right-hand side, in a cell that is migrating through the collagen, you can really see how the nucleus wiggles around and substantially deforms in order to penetrate these small pore sizes inside the extracellular matrix. And we recognize that collagen, on the one hand side, is a very nice substance where it provides a physiological matrix environment, The challenge, however, is though that it's very difficult to control the typical pore size. So in the images I showed you before, we couldn't even see the pore sizes, but you can easily imagine you would get a very heterogeneous distribution, which we can modulate somewhat by changing the collagen concentrations, playing with crosslinkers, or the polymerization temperature, but it's still a very poorly defined physical environment. And so what instead we've done is we've taken advantage of microfluidics and some microfabrication techniques similar to what you do in the generation of microchips, so that we could just engineer environments for cells that mimic the physiological spaces that cells would encounter in vivo. And so we can additionally add, for example, chemotactic gradients that motivate cells to move from one side to the other side. And then when we could create little obstacle courses for cells, essentially, that mimic these geometries cells would find in vivo. And we can have constrictions that are anywhere in size from about 2 micrometers to 15, 20 micrometers wide. Keep in mind that in all of these cases, the cells are vertically confined. So there is a bottom and a top there. So they cannot just go up or down, and they're really sitting in a 3-dimensional environment. And so the morphology is very similar to what you would find in migration in vitro or in collagen gels. You can see the physical size here of these devices. So we can mount multiple of these devices on a microscope slide and then image them on inverted microscopes just to image the cells. And so this is what it looks like. And I think you can clearly see immediately the effect of different spaces and different pore sizes on the ability of cells just to migrate through these environments. In all 3 videos are identical MDA MB231 breast cancer cells that are expressing fluorescent labeled nuclei. And on the left-hand side, we have little just PDMS pillars. The spacing between them is about 15 to 20 micrometers in size. So it's very easy for the cells to navigate. In the middle, we have a higher pillar density, which means that the pore sizes in between are on the order of about 3 to 5 micrometers. So the nucleus already has to deform substantially. And on the right-hand side, the pore sizes are only on the order of about 1 or 2 micrometers wide and 5 microns tall. And you can see really on the left-hand side with the large pore size, it's very easy for the cells to move around. Within about 12 hours, they've completely transversed this obstacle course and made it through these constrictions. In the middle, you can see that the cells are still able to move through these openings, but they take substantially longer than those cells on the left-hand side. And on the right-hand side, you see with the very small constrictions, while cells are able just to enter some of the spaces, they're unable to go very far. So we really see a very clear role that how the pore size determines the ability of cells just to move through these environments. And to show you that it's really the nucleus that impedes the behavior when cells encounter small constriction. This is just a close-up of a cell. In this case, it's a fibroblast, but we see very similar behavior in other cells as well, where the cell encounters the constriction, and you see then it takes substantial time. If you look at the time scale here, it takes several hours for the cell just to generate enough force just to move the nucleus through the constriction. You can see that the cytoplasm already continues to move through it because it can remodel very easily, but it takes a long, long time for the nucleus to pass through the constriction. And just to really drive home the point that even though these devices are just engineered devices, and they look maybe somewhat artificial, but they do often resemble really what we can find in vivo. And so these are some images provided by Peter Friedel's group, just intravital imaging from the skin of a mouse, looking at melanoma cells migrating through the skin. And you can see, in this case, labeled in yellow, nuclei of these cancer cells as they're squeezing through 2 adipocytes. And you can see very similar behavior, how the nuclei are being squeezed to move through this physical constriction. But you would also see very similar effects during intravasation, extravasation, or just cells moving through, for example, just the extracellular matrix inside tissues. And so as you can see, I think from these movies, it's just very clear how we have to take into account nuclear deformability as a rate limiting factor when cells migrate through tight spaces. And this is work that we had started to show that, for example, neutrophils, which have highly lobulated, highly deformable nuclei, and that those properties indeed enable them just to be so good at just infiltrating tissues, but it also plays a role just in generally in other cells as well. There's been similar work by Peter Friedel's group. Dennis Discher's group had similar observation as well as the groups of Mathieu Piel and others. So at this point, we really know that if we are looking at cell migration in three-dimensional environments at pore sizes that are smaller than the size of the nucleus, that we do have to take into account the nuclear deformability just to see how well cells are able to move through these environments. There's a number of other questions, though, that of course now arise from this. One question is, and that is something that we are still working on, as well as a number of other groups, is how are cells able to actually squeeze the nucleus through these tight spaces? Do they pull on the nucleus? Do they push on the nucleus? We've published some work with Steve Rosenfeld that has shown, for example, an important role of non-muscle myosin 2A and 2B in the ability of cells to move through tight spaces. And that is, for example, particularly important in glioblastomas, where again, cells navigate environments with very tight spaces. But we also recognize that cells likely have multiple mechanisms which they can move, and they can both pull and push the nucleus depending on the circumstances. There's been recent papers, two of them just coming out earlier this year, showing that cells can sense the physical confinement, and that indeed the deformation of the nucleus, which can affect the stretch in the nuclear membrane, the tension in the nuclear membrane, that by itself can now activate actomyosin contractility and further modulate the movement of the cells. However, what I want to focus now on the next part of my story is just to see, do metastatic cancer cells have more deformable nuclei? So where we know cells that are highly invasive and that they frequently have to encounter these constrictions, have they maybe undergone some adaptation or some selection in order to particularly be well suited for this task? And so this is work that was done by a former postdoc in my lab, Emily Bell, who now has a faculty position at Penn State University, and we continue to collaborate on this work. And so what we set out to do is just initially really just compare a large panel of different breast cancer cell lines with different metastatic potential just based on their previous characterization in the literature, both in terms of in vitro assays as well as injection into mice, and to see how likely they are to metastasize. And what we recognized, in order to really compare large panel of these cells, we needed to have better tools available just to measure, for example, the nuclear deformability, because current techniques such as atomic force microscopy or micropipette aspirations are very low throughput assays where you can measure one cell at a time. It probably takes you 5-10 minutes for one measurement. You move on to the next cell. And so that's not very suitable to look at large panels, but it's also not suitable to identify, for example, subpopulations of cells. And so we've developed microfluidic devices that function similar to a traditional micropipette aspiration device, where you can see you can flow cells in these devices. Individual cells land in these pockets. Inside the pocket now there's a pressure gradient from the right to the left, which now moves and pushes the nucleus through the small constriction here. And you can easily imagine, if you have a relatively rigid nucleus, that it's not going to move very much into this small channel, and it takes a long, long time to deform. And so this is something that you see here in the bottom curve. On the other hand, if you have a highly deformable nucleus, you can imagine that the protrusion will become much longer and will deform much faster. And so this is what you see in these cases here, shown in red. And what we notice depicted here is just a representative panel of a few small number of cell lines. But when we sorted it, we actually found that it was indeed that the highly aggressive breast cancer cell lines had much more deformable nuclei compared to the less invasive breast cancer cell lines. And not surprisingly, when we then looked at their laminin levels, we found that the highly aggressive breast cancer cell lines that had the deformable nuclei had a reduced expression of lamin A and C, which, just to remind you, these are the proteins that really determine the deformability of the cell nucleus. And so we see that the less invasive breast cancer cells had high levels of lamin A and C, while the more invasive breast cancer cells had very low levels of lamin A and C. And so we've seen this in a much larger panel of cells as well. In order to show that it's really lamins that determine it, we try to see, well, can we now modulate the levels of lamin? Can we take, for example, highly aggressive MDA MB231 breast cancer cells that typically have low levels of lamin A and C and reintroduce an ectopically expressed lamin A to give them stiffer nuclei, more similar to what we've seen in the less invasive cancer cells? And what we do see is initially the MDA231 cells are highly and basically they're very good at moving through these tight constrictions. On the other hand, when we now overexpress laminin A and it gives them more rigid nuclei, many of these cells will now become stuck at these constrictions, as you can see here, where a lot of the cells are piling up in the first row of the constrictions and have a difficult time passing through it. We can also measure, for example, the transit time, how long it takes a cell to move through the constrictions, and we see that the cells that are overexpressing laminin A take substantially longer to move through these constrictions. We see this in the MDA MD231 cells. We see it in another breast cancer cell line as well. So this is something that clearly is not unique to the cells. We've done similar experiments as well using isogenic progression series and find very similar results as well. Conversely, we can take cells that have, for example, higher laminin levels, and we can then knock down laminin A and C, and we show that the reduction of the laminin A and C levels make these cells more invasive. And in this case, this is a collaboration with Katharina Wolf and Peter Friedel's group, looking at fibrosarcoma cells, H1080 cells migrating through a collagen matrix. And if we deplete lamins A and C, so we reduce the lamin AC levels and give them more deformable nuclei, that will substantially increase the migration speed. So these cells will be faster of moving through these constrictions. Importantly, of course, we recognize cell lines are informative, but they don't necessarily reflect everything that was happening inside patients. And so what we wanted to do is we wanted to test whether we can also detect altered lamin levels in patient samples with breast cancer and whether the changes correlate with disease progression. And so we were fortunate to be able to work with Paul Spahn's group at the University of Nijmegen and Radboud University Medical Center, who had already collected a large number of patient samples, including a large fraction of triple negative breast cancer, so highly aggressive breast cancer cells. And so they've created tissue microarrays, and the patients had been followed up for at least 5 years, many of them 10 or 15 years. And so what we were able to do then to use these microarrays and then just stain them for lamin A and C, process them in a blinded way, and then afterwards just send our results to Paul Spahn and have him unblinded and see how do lamin levels correlate with the disease progression. And what we found when we unblinded was that indeed that patients who had low levels of lamin A, and we just normalized it to the lamin Bs just as a staining control, so that patients who had low levels of lamin A had a significantly reduced risk-free survival. So they were at much higher risk of developing metastasis than patients who had high levels of lamin A. And so that definitely correlated well with what we'd seen in the migration assays. At the same time, we know that lamins are not only important to regulate the stiffness of the nucleus, but they play important roles as well in transcriptional regulation. They interact with several transcription factors. They play important roles in chromatin organization. And so we wanted to see, do these changes in lamin levels also affect other cellular functions in these tumor cells? And so what we did is we collaborated with Marco Smolker group here at Cornell, who's a proteomics expert using mass spec. And so we used then stable isotope labeling with amino acids and cultures, a SILAC assay, where we took BT509 cells that are highly aggressive breast cancer cells that have normally low levels of lamin A. And then we ectopically expressed lamin A just to increase lamin expression by about 30 to 50% to get lamin levels similar to those levels seen in less invasive cells. And then we performed proteomic analysis just to see, well, just by boosting the lamin levels about 30 to 50%, how much do we perturb other proteins and which other proteins do we perturb? And what we found was that even this very modest change in the expression levels of lamin A and C resulted in more than 90 proteins that showed a more than twofold change in these cells. And if we break this down into different groups, we see that many of the proteins that were now differentially expressed were involved, for example, in cell metabolism, in stress responses, adhesion and migration, in remodeling the extracellular environment, as well as stress responses, protein homeostasis. And so what we believe is happening, that when cells change the expression levels of lamin A and C, that on the one hand side will have a direct biophysical effect where we have more deformable nuclei, which will make it easier for the cell just to migrate through tight spaces. On the other hand, we also think that it will have, through the proteomic change, a substantial effects on other cellular functions, such as cell matrix remodeling, cell adhesion. We see distinct effects on proliferation as well, and we suspect that there could even be effects on the epithelial to mesenchymal transition as well in these cells. So this was our work on trying to understand really just how do changes in the nuclear properties can dictate behavior of the cells. On the other hand, we know that clearly the nucleus is not just a passive large cargo, but we do have the DNA inside it. And so we wanted to see what happens if we now actually conversely, the physical stress involved in deforming the nucleus as the cell moves through the constriction, how does that affect the integrity and the function of the cell nucleus? And one of the stunning findings we had, which originally was just completely by accident, where we had taken GFP with a nuclear localization signal just to mark the nuclei, just to make it easier to track the cells. And what we noticed is as cells migrated through the constriction, that the nuclear envelope ruptured. So all of a sudden you see here on the left-hand side that the NLS GFP signal starts spilling out of the nucleus. Let me see if the movie stalled slightly, if I can get it back. So as you can see here on the left-hand side, we can have as the cell moves through the constriction, the NLS GFP spills out of the nucleus, spills into the cytoplasm, but then the nuclear envelope is repaired. All of the GFP goes back into the nucleus, and then the process repeats itself at the next constriction. And so again, we see nuclear envelope rupture and nuclear envelope repair. If you see initially, you can even see just the formation of a bleb where part of the nuclear membrane bulges out, then ruptures, and then essentially the NLS GFP can spill out. To show that this is really just a localized rupture event and not just a general disintegration of the nuclear membrane or maybe an opening of the nuclear pores, we developed a fluorescent reporter just to detect the site of nuclear envelope ruptures. And this fluorescent reporter is just based on the cytoplasmic DNA binding protein cGAS, which we fused with a red fluorescent protein. We mutated its signaling domain so that it does not activate any inflammatory pathways and expressed it in cells. And what you can see, what is happening as the cell now moves through the constriction, the nuclear envelope ruptures. As the nuclear envelope ruptures, the genomic DNA becomes now accessible to the cytoplasmic DNA binding protein. And so you can see the accumulation of this NLS, of the cGAS RFP at the site at the nuclear envelope rupture. So you can really see that the rupture occurs at the site where initially the bleb was formed. So it's a very localized rupture. We've done some super resolution studies and other studies. So we suspect that the holes that form are on the order of about 100 nanometers or smaller. Initially, we were just a little bit worried just to say, well, how physiological is this? Does this only happen in microfluidic devices where cells move through relatively rigid PDMS environments? And what was very reassuring to us was then when we repeated the cells in collagen gels, we found very much the same results. And what we found in the PDMS devices, but then also in collagen, is that we get this very strong size dependence. So the smaller we make the constriction size, whether it's using collagen or whether it's using the radiation devices, the higher the incidence of nuclear envelope rupture. So if you have very large pores, you do occasionally observe nuclear envelope rupture, and it's been shown that that still happens because the actin fibers will often compress the nucleus, but it's the smaller you make the constrictions, the higher the incidence of nuclear envelope rupture. If we make very small constrictions, almost every single cell will undergo nuclear envelope rupture, and we see an almost identical behavior whether we have cells migrating through the migration devices, shown here on the black line, or whether cells migrate through the collagen matrix, indicated by the red line. So it really seems to be a very universal mechanism. And what's even more convincing to us was then when we sent some of our cells with these fluorescent reporters to Peter Friedel's group just to perform intravital imaging in vivo studies, they observed the very same behavior. So these are H1080 fibrosarcoma cells migrating now inside the tissue of a living mouse. And again, you can see the formation of the bleb at the leading edge of the nucleus. You see the nuclear envelope rupture, NLS GFP spill out. So the next time spilling out, and then within about 30 to 90 minutes, the nuclear envelope is repaired, and then the NLS GFP is being re-imported into the cell nucleus. we were able to perform a lot of detailed studies just to understand the process of the nuclear envelope rupture and repair in more detail. And so what we found is what is happening, and this has been subsequently confirmed by a number of other groups as well, that it likely starts off with a small defect in the nuclear lamina where you have now the nuclear membrane is not being supported by the underlying nuclear membrane. And as the cell is squeezing through the constriction and exerts a lot of pressure on the cell nucleus, which increases the hydrostatic pressure inside the nucleus, you have now at these sites of these local defects in the lamina, the nuclear membrane starts blebbing out. The bleb increases as fluid flow goes into the bleb. If the size of the bleb becomes large enough, you can often see chromatin entering the bleb as well. And so it starts looking almost like a micronucleus, but it's still connected here. And then lipid membranes typically cannot expand for more than a few percent, and so they can only be strained very little. And so what happens that ultimately the nuclear membranes rupture, and so we have now an uncontrolled exchange between the nuclear interior and the outside. The inner and outer nuclear membranes will rapidly reseal, but it still leaves open a channel here that still continues to allow uncontrolled exchange between the nuclear interior and the outside. And then eventually what you'll have is that escort proteins, particularly escort 3 proteins, are being recruited to the side, and they help then now just tie up and to seal the rupture here, the rupture site. And so within about 15 minutes, typically you'll have the nuclear pore, you have the hole in the nucleus repaired. And often what we find will actually have an accumulation of lamins there. So where there's lamins are being recruited to the side, and what we often call is a lamin scar. So you have an increased amount of lamins at the side at the former rupture. And so we've identified a crucial role of CHIM7 and escort3 proteins. Similar work was done at the same time, published by Mathieu Piel's group. And then recent work has also shown that in addition to the escort proteins, that BAF and LEM domain proteins play a crucial role as well in the repair process itself. What was intriguing to us that the cells survived the nuclear envelope rupture and were able to repair the nuclear envelope and then keep going. On the other hand, what we noticed, particularly when we stained the cells for gamma H2X, that a lot of cells were positive for DNA damage. And then we've performed live cell imaging using fluorescent reporters to detect double-stranded DNA breaks, such as in this case 53BP1 labeled with an M cherry. And so what you can see on the left-hand side as the cell moves through the constriction, you see the nuclear envelope rupture, but you also see now these red foci appearing, so indicating sites of DNA damage in the cell. And we again could show that this behavior was very much dependent on the size of the constriction. So the smaller we made the pore, the higher the incidence that nuclear envelope rupture occurs. What's intriguing though, we wanted, and so that I should point out, so this is something we'd seen in our studies, but very similar results were also obtained at the same time by Mathieu Piel's group, and then subsequently by Dennis Disher's groups as well. So where we recognize if cells squeeze to tight spaces, that can result in an increase in DNA damage. What was unclear, though, is exactly what causes the mechanical stress, or how does the mechanical stress cause the DNA damage? And in particular, one question that had been baffling us, and that's something that Mathieu Piel and I had already discussed when we were publishing our papers and we were exchanging our information, is the DNA damage caused by the nuclear envelope rupture, or is the deformation of the nucleus itself, even without any rupture, sufficient to cause DNA damage? And so this is something that we then investigated in more detail using time-lapse imaging, using our fluorescent reporters for DNA damage and for nuclear envelope rupture. And so this was work done by a PhD student who just recently graduated and just moved to Boston last year as well, so Pragesha. And so what she found is that indeed DNA damage can occur even without nuclear envelope rupture, depending on the cell lines. And you can see in this case, this is an MDA-MB231 cells moving through a dense collagen matrix. And you can clearly see the nuclear envelope remains intact, so the NLS-GFP signal stays inside the nucleus, but you can, as the cell moves through these constriction, you see the large nuclear deformation. You can see how these new red foci appear, indicating new sites of DNA damage. And you can see these red dots appearing. So we clearly see evidence that DNA damage can arise just from nuclear deformation, even in the absence of nuclear envelope rupture. What was intriguing to us was that seemed to depend very much on the cell line that we're working with. So if we were looking at breast cancer cell lines, BT549 or MDA-MB231 cells, indeed, most of the DNA damage was caused just by deformation alone. And so in very few cases did we see nuclear envelope rupture, but we still saw lots of DNA damage. In contrast, when we looked at fibroblasts, H1080 cells, or RP1 epithelial cells, we saw that in those cases, most of the DNA damage was actually associated with nuclear envelope rupture. And that is something that we're still trying to understand. What are the difference? Is it intriguingly that these cells, the BT549 and MD231 cells, they are P53 mutant cells. These other cells are P53 wild type cells, but clearly there are many other differences between these cell lines as well that we are still trying to understand. Just to get real confirmation that is it just the deformation that is responsible for the DNA damage, we thought, well, if it's the deformation, then even in non-migrating cells, deforming the cell nucleus should be sufficient to induce DNA damage. And so we worked together with Richard Superfine's group at UNC Chapel Hill and a very talented graduate student there, Chad Hobson, who've designed an atomic force microscope that can provide high resolution 3D imaging of the nucleus as we compress it. And then we use these individual nuclear compression studies just to see whether compression of the nucleus, even without rupture, was sufficient to induce DNA damage. And we can see on the top panel, indeed, that if we compress the nucleus, you can really see the substantial nuclear deformation. We see new foci forming, indicating new sites of DNA damage, while at the bottom, which is just a control condition where we just gently tap the cell, where we just put the cantilever on top of the cell without inducing large nuclear deformation. And we see this cell has a one pre-existing DNA damage foci, but it does not form any additional DNA damage. And so we can quantify this as well and see that if we compress the nucleus, we have a substantially increased rate, an increased probability of forming new DNA damage compared to non-compression. We were interested to see whether some of this could maybe be related to the cell cycle stages at the cells. And of course, we know that cell cycle progression, cell cycle state are very important determinants for the formation of DNA damage. And so what we did is we conducted these experiments now with cells expressing the Fuji cell cycle reporter, in which case, if you're not familiar with it, cells switch between green and red fluorescence depending on the phase of the cell cycle that they're in, where you see if they're in G0, G1, the cells are in red, they're nuclear labeled in red, and if they're in S or G2 phase, they're green. And so that allowed us now, when we combined the Fuji reporter with the other reporters, to detect exactly when DNA damage occurred in the cells. And by combining it with the nuclear rupture reporters, we could also now distinguish between DNA damage associated with nuclear envelope rupture versus DNA damage caused just by deformation without nuclear envelope rupture. And what was intriguing to see was that the DNA damage that was associated with nuclear envelope rupture occurred in all phases of the cell cycle, and consistent with the idea that maybe there's cytoplasmic nucleases, for example, that can get into the nucleus. And then once you breach the nuclear envelope, these nucleases go into the nucleus, they can act on the DNA. And clearly that effect would be independent of the cell cycle. On the other hand, we found that DNA damage that was associated with nuclear deformation in the absence of nuclear envelope rupture only occurred primarily during S and G2 phase of the cell, so during the replication phase of the cell. And so that motivated us to look more closely at whether some of the DNA damage was associated with DNA replication forks. And so we could combine time-lapse imaging and then fixing the cells using immunofluorescence to detect where these new 53 BP1 foci, so where new sites of DNA damage arose. And what we found, a lot of them did occur at sites of replication forks where we used phosphorylated RPA as a marker for single DNA that indicates replication forks. And so we really saw that a lot of these new foci appeared at replication forks. Then we wanted to see, well, does now physical stress increase replication fork stalling, which could provide one potential mechanism for the DNA damage. And so again, we used compression assays paired with immunofluorescence to look at RPA1, which accumulates particularly at stalled replication forks. You get these bright phosphorylated RPA foci. And we could indeed show, if we use substantial compression of nuclei, we could see a lot of the phosphorylated RPA foci compared to controls. And so we saw a significant increase in the MDA MD231 cells, which showed this deformation-induced DNA damage mechanism. In contrast, when we looked at HT1080 cells, fibrosarcoma cells, where most of the DNA damage was due to nuclear envelope rupture and not to deformation, there we saw that the compression did not affect the replication forks at all. And so we did not have any effect of compression on the number of the RPA foci. We saw a very similar effect when we looked at cells migrations instead of external compression using migration. And again, where we saw that in the MDA MB231 cells, if the cells migrated to a very small constriction where the nucleus had to deform a lot, we saw a substantial increase in the stalled replication forks, indicating, in this case, using this PCNA as a marker, that the PCNA signal increases as replication forks stall and PCNA accumulates. Well, if you looked at HT1080 cells, where it's likely the DNA damage is caused by nuclear envelope rupture, we saw no effect of compression or on the moving through a small constriction on the PCNA foci. And so in conclusion, and since this is published work, I'm not going to go through all of the details. What we've recognized is that mechanical stress on the nucleus and deformation of the nucleus can cause DNA damage through two likely distinct mechanisms, although there might be some overlapping aspects in it as well. So both confined migration or external cell compression can cause DNA damage. And the DNA damage can either be associated with nuclear envelope rupture, in which case it can occur through all stages of the cell cycle. And as I mentioned, one of the hypotheses is that it's likely associated with the influx of cytoplasmic nucleases, although it has also been speculated maybe whether efflux of nuclear DNA damage repair factors could contribute as well. In contrast, if you look at nuclear deformation associated DNA damage, that only happens during the replicative phase, so during SNG2 phase. And what we think is really happening, that you have an increased replication stress, you have replication fork stalling, and that could be, for example, because the deformations twist and results in torsional stress on the DNA that promotes the replication stress, but it could also cause other conformational changes in the DNA that are being recognized by nucleases that act on them. I should point out, while in the previous parts I pointed us to localize double-stranded DNA breaks. What we noticed, though, we can also see much more substantial damage to the nucleus as the cells move through these constrictions. And in this case, we see really severe signs of nuclear fragmentation, where we have cells that start off on the left-hand side having the expected nice round oval nuclei. And as they move through the constriction, you will see how small pieces of the nucleus almost get fragmented off and get pinched off as the cells moves around. You see it after the first constriction, but you see it really particular as the cell have moved through repeated constrictions. And you see many of these fragments forming. In some cases, they're still attached to the primary nucleus. In some cases, they appear to almost just completely pinch off. And what we found was that these fragments are very often positive for gamma H2X, very similar to what David and others had shown for micronuclei. And so we think really the fragmentation of the nuclei, this is potentially a mechanism that even dramatically increases the amount of DNA damage just by pinching off cells, causing DNA damage. And then when the cells get reincorporated after mitosis into the cells, then you might have really chromothripsis and very dramatic effects on the genomic integrity. And so I just showed you before the representative video. Here you can see just the quantification. So we see this effect both in the constriction channels in our microfluidic devices, that cells that have moved through the devices have an increased number of fragmented nuclei that seems to persist afterwards. But we see the same effect as well when cells move through collagen matrices, and the effect becomes more pronounced if we use collagen matrices with smaller pore sizes, or if we prevent the cells from degrading the collagen matrix, and so that they have to really move through existing pore sizes. And so that is something where we're right now starting to look at more at the long-term consequences. So rather than just seeing what are the acute DNA damage, but to see are cells accumulating more and more DNA damage over time as they move through such confined environments. Lastly, and this is the last topic I have, we wanted to see, in addition to really these genomic changes and DNA damage, can we also see changes in the chromatin organization of nuclei? And so this is that Jeremiah Shah described as a PhD student in my lab, where he noticed when he stained cells for heterochromatin or euchromatin markers, in particular looking at H3K9 triple methylation and H3K27 triple methylation as markers for heterochromatin, and then we normalize it to the euchromatin marker H3K9 acetylation, just to make sure that we're truly looking at chromatin modifications rather than just a physical compression of chromatin. Because you could imagine if you were just squeeze chromatin together into a smaller space, then all of the existing chromatin marks should increase. On the other hand, if we have true modification, you'd expect to see, for example, an increase in the heterochromatin marks and a decrease in the euchromatin marks. And so that's what we typically do, is we double stain and then we look at the ratio. And so what we see here in these images is really this dramatic increase in the heterochromatin marks as the cell moves through these constrictions. And you can see some of the increase even in the wider channels that are 15 microns wide, where the nucleus doesn't have to deform as much. But keep in mind that the channels are only 5 microns tall, so the nucleus is still being squeezed. And then we see much more dramatic changes in the smallest channel. And so these are the representative images, but I think you can see it more clearly when we quantify it. And so what we express as the normalized ratio of, for example, H3K9 triple methylation. And you really see, while the controlled channels, we see an increase in the heterochromatin marks as the cells move from before the devices through the first constrictions and come out of it, where we see the substantial increase. But this increase is even more pronounced if you look at these small constriction channels that are less than 2 microns in size. And this applies both to the H3K9 triple methylation as well as the H3K27 triple methylation. And we do see similar results as well if you look at DNA methylation as well. So very consistent findings there. We really see a progressive increase. So if you look at the events to individual channels, we see it does increases over time as the cell progress through these constrictions. Importantly, we see the same effect as well in collagen. So this is, again, it's something that it's not only found in microfluidic devices, but we can see the cells move through collagen as well. And again, we see the more the cells have to deform through the collagen, the more of these heterochromatin marks we see. And so the stronger the increase in the histone modifications. In terms of trying to understand the mechanism, it's good to understand in terms of what enzymes control the histone methylation modification. And so the key enzymes, if you're not familiar with them, there's histone deacetylases that are important just to remove acetyl marks from the histones, and then histone methyltransferases, which now add the methyl group to these groups, for example, to the H3K9 and H3K27. And then, of course, there's histone demethylases that can now remove the methyl groups, and then we can have histone acetyltransferases that, again, later on add the acetyl marks to it. But so we focused on the histone deacetylases and the histone methyltransferases to really show that these modifications are enzymatically modified. And so we could see if we treat cells with the histone methyltransferase inhibitor, in this case DZNAP, for example, we can significantly reduce the amount of the heterochromatin formation as the cell moves through the constrictions. We don't completely abolish it, but we significantly reduce it. But you'll see if we combine it now, combine treatment of both the histone methyltransferase inhibitors and the histone deacetylase inhibitors, then we essentially completely abolish any of these chromatin modifications, indicating really that these are true enzymatic changes that occur as the cell moves through the constrictions. I don't have time to go into all of the details. We've done now some ATAC-seq studies just to look at where specifically these changes occur. Interestingly, a lot of them occur at intergenic regions, particularly at the centromeric and telomeric regions, where it appears to be an expansion of existing heterochromatin rather than completely new modification of new sites. And we do see that there's some sites where we increase the chromatin accessibility, so where there's reduction in the heterochromatin, and those sites typically occur at promoter sites as well. And so we do expect to see some changes in gene expression as well. in terms of understanding the mechanisms, we do see a role of stretch-sensitive ion channels. And so this is a topic that has recently come up increasingly, where nuclei and the ER that's close to the nucleus contain stretch-sensitive ion channels. And the idea is that by stretching the cell and stretching the nucleus, these channels can open up, and a lot of these chromatin modifications then appear to be sensitive to calcium influx through these stretch-sensitive ion channels. And so that is something that we are investigating in more detail. Importantly, though, we want to see, well, what is the relevance of these heterochromatin changes? And so we wanted to see, well, what happens if we prevent these changes? And so we've treated cells with the histone methyltransferase inhibitors and then studied their ability just to migrate either in collagen or through the migration channels. And what we found was that if we block the histone methyltransferases, so we prevent the induction of this heterochromatin, we significantly impaired the migration of cells. So if you're looking at the transit time, how long it takes a cell to move through the constriction, it takes a cell substantially longer, even through the larger openings, but the effect is even more pronounced when the cells move through the smallest sizes. So we really think the effect of the heterochromatin modification enhances the ability of the cells just to migrate through these three-dimensional environments. We are currently testing the hypothesis that it might also help to prevent DNA damage, for example, and so might play a protective role as well. But that is something that we're still investigating in more detail. And so just to come to the end, so hopefully I've been able to convince you and show that really the nuclear deformability plays a crucial role during cell motility in three-dimensional environments, and that reduced laminin levels promote cancer cell invasion by not only increasing nuclear deformability, but also by promoting metastasis likely through additional pathways, just by altering expression of other key proteins. And that physical stress during the migration can result in nuclear envelope rupture and DNA damage, and that the DNA damage can arise from nuclear envelope rupture or from nuclear deformation alone, with nuclear deformation increasing replication stress. And I should point out that this, while we mostly looked at it in migrating cells, but we suspect that this could also play a role in physiological settings where cells are just being compressed without migrating, as we've seen that the nuclear deformation alone is sufficient to induce this DNA damage. And what we think is that this mechanically induced DNA damage may further contribute and add to other mechanisms that increase the genomic stability in cancer cells. And then intriguingly, in addition to these genomic changes, we also have these epigenetic changes, particularly the increase in heterochromatin, that we're still trying to understand in more detail exactly what is the role, and is it more biophysical changes that we change the size or the deformability of the nucleus, or is it more that in the end we change the gene expression, which then promotes the ability of cells to migrate around? With that, I'd just like to thank the people in the lab who've done all the work, a number of former people and current students, many collaborators who've been involved in the studies. I think the slide is too small to add all of them here. And then our funding agencies. And with that, I'd like to thank you for your attention, and I'm happy to take questions. Thank you very much.
Dr. Pellman, are you there?
I think it's just muted.
Okay. At this time, if you'd like to submit any questions, please do so in the Q&A tab at the bottom of the screen. And we will present them to Dr. Lammerding. Thank you, Dr. Lammerding, for a great presentation. Okay,
thank you.
Here he is.
Sorry about that.
No problem.
I wasn't allowed to unmute myself.
Oh.
Well, let me get things warmed up with a couple. I have a lot of questions. So one question I have is, what do you think is the source of variability in lamin A expression across a given tumor type?
There's probably a number of possible explanations. There have been some previous studies that showed that, particularly in lymphomas, for example, which often have low levels of lamin C, that there's promoter methylation that plays a role and that could explain some of the variability. That study did not find changes in the promoter methylation in breast cancer cells. And we've done some preliminary studies on it. We see that AKT and PI3 signaling appears to be a role, and so that connect both on the transcriptional regulations of lamin AC as well as the protein stability, the turnover. And so we've seen if we used AKT or PI3 inhibitors that we can modulate the expression levels of lamin AC in some of the breast cancer cells. So I think that is probably one pathway. I suspect there might be additional pathways, and it might vary from tumor to tumor, whether in some cases it's transcriptional, whether in some of it's post-translational. Mutations don't seem to play a major role, so it's relatively rare that you find mutations in lamin proteins. In most cases, those seem to be more likely passenger mutations rather than really driving mutations.
Okay. That's great. And then I wondered, for the DNA damage that's happening during migration through constriction that's unrelated to envelope breakage, have you looked at various topoisomerases and their role in possibly generating the damage?
So that is something that we've started to look at it. So I think we've tried topoisomerase inhibitors. The tricky part is, of course, then if you inhibit them, you just get at baseline already a lot of DNA damage itself. So we've been started to do some experiments trying to see if we can overexpress topoisomerase 2, for example, and to see if that can reduce the DNA damage. It's been technically a little bit challenging just based on the size of the topoisomerase and getting that into the cell, but that is something that we suspect is more informative. We had hoped that maybe just using partial inhibition of the topoisomerase that we could sensitize cells, but it seemed to be very difficult just to find a threshold where we just didn't already, by inhibiting it, cause massive baseline DNA damage.
Okay, and I'm just taking my host prerogative here. When cells are going through a constriction and you get the changes in the epigenetic mark, it looked like that was happening globally across the whole nucleus. It's not a local phenomenon restricted to the constriction site, or is there any bias?
It's hard to tell, since we, for the most part, the epigenetic changes we have to do using fixed cells and just stain the cells with antibody against the histone modifications. And so we can't, or it's difficult to observe it in live cells. So we typically look at cells that have gone through it. And so we see that it accumulates over time. And so then, of course, the entire nucleus has passed through it. We haven't tried to see if you could just deform part of the nucleus and if you'd only see changes there. But I suspect that these changes are more global. We've seen increase in HDAC3 activity, for example. And again, I suspect once you increase the activity, that could act globally. And at least when we, based on our ATAC-seq studies, we really saw that a lot of the increase in the modification or the decreased accessibility was fairly global, as is particular in intergenic sites, so at the centromeres and at the telomere regions.
Okay, great. Thank you very much.
And I see here from Stamatis. So he is asking, are these epigenetic changes transient or persist even after cells migrate through the constriction? So we've done experiments where we combine time-lapse imaging and then after 5 days or after a week, fixing the cells, then staining them and using the live cell imaging just to determine when the cell last passed through constrictions. And so we do see that the epigenetic changes persist for at least 5 days after the cell passed through it. Interestingly, they even persist after the cell goes through mitosis. So we had a number of cells that went through the constrictions, divided, and then even after the division, we could see that those cells still had higher levels of heterochromatin than cells that had not gone through the constrictions. We don't know how long they persist. In terms of, technically, we weren't able to follow the cells longer than 5 days. We had done some other studies before where we had cells that had gone through migration devices, collected them, expanded them for about 2 weeks, but of course that's a substantial cell. And then we found at that point we couldn't really detect changes in chromatin levels anymore. So I think the answer is somewhere between 5 days and 2 weeks, although it might depend also on maybe on the rate of division and on other factors as well. I think.
Okay. Unfortunately, it looks like I don't have access. The chat is not showing questions.
Oh, interesting. So I can just read.
So I think so. You'll have to field your own questions from the
Sure,
chat.
no problem at all. So I'll see the second question is from Paranjan Gimiri. Is increase in heterochromatin related to reduced laminin levels? I don't think we compared it there. So I know that the deformation-induced DNA damage could not be explained by difference in laminin levels because it did not correlate with it. For the heterochromatin, we've seen is if we completely deplete lamins AC, we can essentially eliminate the effect, but that's a little bit harder to interpret because we know that overall lamins are important in heterochromatin formation. And so if we delete lamins, you already lose a lot of heterochromatin. And so we do see deletion of lamin AC abolishes much of the effect, but we don't know if that's specific to the migration-induced effect or that overall you need lamin AC for some of those changes. We do see some effect of Amarin on it as well, but that's something where we still need to develop a better picture. I think these are the questions I see here so far. Let me see if there's.
Yes, those were all the questions that were submitted. So we want to thank you again, Dr. Lamerding, for a wonderful presentation. And thanks again for your time and effort. I will be in touch with you via email for final remarks. And thank everyone for tuning into this week's seminars in oncology. Okay, bye everyone.
Okay, thank you very much, everyone. Thank you for hosting.
Bye-bye.
Bye-bye.